Bismillahirrahmanirrahim. Good morning, good afternoon, good evening, wherever you are joining us from. I'm Dr. Massab Umair, working as a senior scientist and in charge of virology department at National Institute of Health, Pakistan. It is my pleasure and privilege to moderate today's session, which is titled as MERS coronavirus, a potential threat to global health. I'm truly honored to have as our speaker a true pioneer in the field of virology, Professor Joseph Sriyal Malik Peiris. Professor Malik is the chair of virology at the School of Public Health, the University of Hong Kong, and co-directs the WHO H5 reference laboratory and the WHO SARS-CoV-2 reference laboratory at the University of Hong Kong. With a career spanning decades, he has made seminal contributions to the field of clinical and public health virology, particularly in emerging viral diseases at the animal-human interface using a One Health approach. In 2003, Professor Malik and his team discovered SARS coronavirus, the novel coronavirus responsible for severe acute respiratory syndrome. His research has since encompassed coronaviruses such as SARS coronavirus 1, MERS coronavirus, and SARS coronavirus 2, as well as human and animal influenza viruses like H5N1, H9N2, and H7N9. He has served on WHO missions to Saudi Arabia on MERS and to China on avian influenza H7N9, bringing his expertise to global health challenges. Professor Malik has published over 800 peer-reviewed articles, cited more than 76,000 times, and has received numerous prestigious awards, including the Royal Society Fellowship, the John Dirks Canada Gardner Global Health Award, and the title Officier de la Lion d'Honneur from France. He is a global leader in virology, a champion of One Health concept, and a guiding force in combating emerging infectious diseases. It's my absolute pleasure to welcome Professor Malik. Sir.
Thank you very much, Massab, for your invitation to talk with your group. It's a pleasure to do so. So I believe, as I hope I can convince you, that this is a significant threat to human health going forward, and a potential pandemic threat as well. So just to remind you about coronaviruses, as you know, from SARS coronavirus 2, they are positive sense single-stranded RNA viruses, the largest RNA virus in genome size. And we have 4 structural proteins, the spike, the M, N, and E. Now, unlike other RNA viruses like influenza and HIV, coronaviruses have no proofreading mechanism, do have a proofreading mechanism, whereas these other coronaviruses like influenza and HIV do not have this proofreading mechanism when the virus replicates its RNA. And because of that, the mutation rate for coronaviruses is lower than that of other RNA viruses like influenza or HIV. However, there is the possibility of recombination and deletions and sometimes insertions, which allow coronaviruses to also have a fair bit of genetic plasticity. Coronaviruses infect a large range of mammalian and avian species, over 4,000 coronaviruses in bats alone. And they are important diseases in humans and domestic livestock. So this shows a phylogenetic tree of the relationship of the different coronaviruses. And let me get my pointer. So you have the alpha coronaviruses down here, and two of the endemic seasonal coronaviruses in humans, 229E and NL63, are found in the alpha coronavirus group. And you can see these stars here. This is one is a feline canine recombinant coronavirus, and here is a porcine delta coronavirus. Now, both these coronaviruses are causing zoonotic spillovers to humans, and they are potential problems for the future. Then we have the beta coronaviruses, which encompass a very large group of viruses. And here you have the OC43 and HKU1, which are also seasonal coronaviruses and endemic in humans. And here in the sarbecovirus group, you have SARS coronavirus 1 and SARS coronavirus 2, which cause COVID-19. And another group are the merbicoviruses, which includes MERS coronavirus. So you can see that MERS coronavirus is quite distinct from SARS-CoV-2 or some of the other human coronaviruses that are endemic in humans. So all of you are familiar with SARS coronavirus. Its reservoir is in bats, and it jumped to humans maybe through an intermediate host. And we are very familiar with the impact that it caused globally. However, as I mentioned, we do have, before SARS-CoV-2, we had 4 other coronaviruses that were endemic in humans. NL63, 229E, OC43, HKU1, generally causing mild upper respiratory infections, but occasionally causing serious disease. But the important point to note here is that at least 2 of these endemic human coronaviruses spilt over to humans relatively recently. So OC43, on the basis of genetic analysis of the virus and animal coronaviruses, very likely spilled over to humans from a bovine source, because there's bovine coronavirus very closely related virus. And if you date this phylogenetically, this is just only about 120, 150 years ago. And presumably at that time caused a pandemic, because now this virus is present everywhere in the world. And similarly, 229E, we believe it spilled over to humans from camels or alpacas or related species, the camelid species, because there are very closely related viruses in camels. And this happened maybe 300 years or so ago. So the point I'm making is that in addition to SARS, there have been, over the last 500 years, a number of other coronaviruses that have spilled over to humans and caused pandemics. And of course, they are now endemic with us. Then, of course, we have SARS coronavirus 1, which caused problems in 2002, 2003, and MERS coronavirus, which I'll talk about. And I did mention these other zoonotic spillovers that are taking place right now. So there's this porcine Delta coronavirus, which is causing human disease from time to time. It has not got established in humans. It is not transmitting human to human. And there's this canine feline recombinant coronavirus, which is also spilling over. So these are potential threats as well. But I will focus on MERS coronavirus from here on. Now, MERS coronavirus was first discovered in 2012 by Dr. Zaki. He's an Egyptian virologist at that time working in Saudi Arabia. But after, and that was late in 2012 from a patient who had severe pneumonia and who died. But after the virus was discovered and the genome was available and the serology tests and the PCR tests were available, it was found that a previous outbreak that occurred in Jordan involving 6 individuals that same year was also caused by MERS coronavirus. And after that, with global awareness and testing, you can see this WHO epidemic chart or outbreak chart, and you can see outbreaks, infections, zoonotic spillovers, and outbreaks from time to time. And the important thing is that these have spilled over from, as I will show you, from camels to humans, but there has also been human-to-human transmission. For example, in this outbreak in South Korea, which was initiated by a returning traveler who had traveled in the Middle East and come back to South Korea, and he then started an outbreak, of course, accidentally in South Korea, which led to 186 cases. Very clearly, you can see that that outbreak went from human to human to human to human, right? So fairly long chain of human transmission. And similarly, some of these outbreaks in Saudi Arabia have also gone human to human to human, at least within healthcare settings. But finally, these outbreaks have been able to be controlled. But this is the concern. So it is a virus that does have the capacity to transmit human to human when the conditions are right. And of course, with additional mutations and changes, it may acquire capacity for sustained human transmission, which is what leads to a pandemic. So if you go back to 2012, after the first cases were reported in Saudi Arabia and Jordan, the immediate question then was, what was the zoonotic source of human MERS coronavirus? And there were lots of investigations done. And for example, there was this paper that came out saying that from a particular study that they found a MERS coronavirus-like virus in bats. But subsequently, it is shown that this particular study is flawed, because very likely the PCR signal they got was cross-contamination. Although, of course, the natural reservoir is likely to be bats, the MERS coronavirus that is spilling over to humans is coming from camels. And where camels got it from ultimately may have been bats or something else, but we'll talk about that in a minute. So it was in 2013 that a number of studies, initially the one from Marianne Koopman's group in Erasmus, and then from our own group, the Marianne Koopman's looking at camels in, well, both Marianne and ourselves, we were looking at a whole range of animal species because we had developed serological tests to look for antibodies. She found evidence of antibodies in dromedary camels in Canary Islands, and also in Oman. In our own studies, we found evidence of antibody in camels in Egypt, and then later in Saudi Arabia as well. And of course, subsequently, many groups have found evidence of virus RNA as well in camels. So these are some of the early studies from our own group were suggesting. So we had by this time developed, of course, microneutralization tests, but also a pseudoparticle neutralization test, which can be done in BSL-2. As I said, found evidence of antibody, very high seroprevalence in dromedary camels in Egypt and in Saudi Arabia. But you might remember there are camels in Australia which were taken there at the beginning of the 1900s from Afghanistan area. But the Australian camels did not have any evidence of the MERS coronavirus antibody. And then we also were able to, in collaboration with our Saudi colleagues, look at camel sera from the 1990s. So that is long before the virus was discovered. And you can see again, so this virus was not newly emerged in camels. It seems to have been there maybe at least for 30, 40 years. And you can see many of the other animal species had no evidence of antibody or very little. Now, we come back to this question where, so it is very clear that the source of human disease are camels, but where did the camels ultimately get it from? And this is a question we still do not know. So this again is a close-up of the beta coronavirus group. So this is the 2C group where MERS coronavirus is. And there are many camel viruses that are fairly closely related, but not very closely related. Okay, so just as an example here, I told you that so this is the seasonal human coronavirus OC43, and you can see bovine coronavirus is almost identical, very, very close to the human virus. This is why we are confident that the humans got infected from a bovine-related virus. But there is no such very closely related virus for MERS, but this is presumably because we haven't found it yet, because there are so many animal species, so many bat species in nature, which we haven't tested yet. Now, it was subsequently found that the receptor for MERS coronavirus in humans is DPP4. And you can see there are some bat coronaviruses which do also bind DPP4 and can be cultured in human cell lines. These are 2 bat coronaviruses here, and also a pangolin coronavirus. You remember that SARS coronavirus 2 also has a related virus found in pangolins as well as in bats. So there are a number of viruses out there, but you can see that phylogenetically, these are not very close to MERS coronavirus. So we still don't know that the ultimate reservoir of MERS coronavirus. Now, coming back to humans, these are data from FAO and WHO. If you look at human cases, some of them have evidence of close contact with camels. So these are these red part of the stacked bar chart. And some of these, the blue ones, have evidence of contact with other MERS cases. So that means this is secondary cases from other humans, all right? But you can see that there is this orange part of the bar where there is no clear source, either camel or human, where they may have got infected from. And in the early days, this caused a bit of confusion as to where these people were getting infected from. But now we know the epidemiology of MERS coronavirus quite well. So as I said, the virus is in camels. It transmits from camel to camel. And when you look at all the camels, almost 100% of them have evidence of antibody, meaning they have been infected at some time in their life. So the virus is maintained in camels. From time to time, it spills over to humans. And quite often, these spillovers to humans are not detected because they may be mild. And it may even spill over from one person to another, which still may not be diagnosed or detected. And it is only when it reaches somebody who is older or who has comorbidities like diabetes, hypertension, obesity, etc., that is when the humans get ill and go to hospital and get diagnosed. So you can see, this is just a hypothetical example, but you can see this particular chain. This is the first person who got diagnosed, but there were possibly a number of other people in the chain between the camel and this particular person who got diagnosed. So this is why you have people where they don't have a clear history of exposure either to camels or to other patients. And then, of course, when these patients enter hospital without good infection control, you can lead to an outbreak in hospital. So you have these large clusters of human-to-human transmission. Of course, the interaction of humans and camels through camel herding, camel slaughterhouses, and in the Middle East, for example, you have camel racing, you have camel exhibitions. I don't believe, I'm not sure, maybe you can educate me, but I don't believe you have camel racing and this type of camel exhibitions in Pakistan. But clearly you do have camel herding and camel slaughterhouses, etc. Now, this is again a different view of the WHO outbreak chart of MERS coronavirus. Now, most of these cases, the zoonotic cases, have occurred in the Arabian Peninsula, in the Middle East, right? Except, of course, travel-associated cases like this orange outbreak in South Korea. But if you look at dromedary camels, you can see that the distribution of dromedary camels is very, very wide. In fact, almost 80% of global dromedary camels are found in Africa. Only 4% are found in the Middle East, and a significant proportion are found in Central Asia and in other parts of Asia as well. They are found in places like Kazakhstan. In Mongolia, they are Bactrian camels, which are a different, the two-humped group. Now, so these red dots that I have put here are basically saying that wherever these camels have been tested, they are seropositive and PCR positive as well. So although most of the human cases are being reported here in the Arabian Peninsula, the virus itself in camels is present in a much wider region in Africa and in Central Asia. So the question then is, why has human cases not been reported outside the Middle East? Now, you can think of a number of possible reasons why. Now, I showed you that infected animals are present, so the virus is present. Now, could it be behavioral factors, the close contact with camels? So we, in fact, looked at this in two studies in Africa, in Nigeria and in Morocco. And essentially, the interaction between humans and camels in Nigeria, Morocco was very similar to what you found in in the Saudi Arabian or the middle or the Arabian Peninsula, except of course you don't have camel racing and these camel exhibitions, but otherwise you have lots of close contact, people using camel milk or raw camel milk, etc. So then the next question is, okay, the virus is present. Now I'm now talking specifically about Africa, which is where we have done most of our studies. Is there evidence of infection taking place? A number of studies done by others and by ourselves have shown that infection is taking place. It is not very common on the basis of antibody testing. So you can see here in Morocco, this is one of our own studies where we had evidence of low prevalence of neutralizing antibody. This is in humans with camel exposure. And here in Kenya, there are a couple of studies which have shown camel handlers and camel exposed individuals having antibody. And this is, again, as I said, neutralizing antibody, very specific. And in a study in Kenya, where they also look for RNA, that is by PCR, in these camel herders, they found some camel herders with evidence of virus RNA as well. So infection does seem to be taking place. Now, you have to also put this into context of how sensitive the neutralization test or any serological test for the spike, which is the specific test that you can use, is in infection. So this is a study we did in collaboration with our colleagues in South Korea during that outbreak. These are patients, PCR-confirmed patients, most of them with X-ray changes in the lungs, confirmed to be MERS. And you can see that the red lines these are the antibody titers, antibody risers, and the antibody level then lasts for a long time. The red cases are the severe cases who are oxygen dependent or in ICU. And you can see they have very strong antibody responses and it lasts for a long time. Now, the green ones are the milder cases who did not require oxygenation, although they did have X-ray changes in the lung. And some of these make very little or no neutralizing antibody or even spike binding antibody. And this is another study that came out just last year, essentially saying the same thing. So, and this is with ELISA, S1 domain spike binding antibody, asymptomatic cases, very little. So what this means is that the seroprevalence data I was telling you is probably grossly underestimating the real extent of human infection. Now, there was another study that in Saudi Arabia done by Stanley Perlman and Jingsun Zhao, looking at confirmed cases, PCR confirmed cases. They looked at antibody and also looked at T-cell responses, and they found, again, some people did not make good antibody response, but they did make T-cell responses. So because of that, we did a study in Nigeria, in a camel laboratory in Nigeria, in collaboration with colleagues there. And first and foremost, we had to validate our assay, T-cell assay. So let's look at this. These are CD4 T-cells. So we have negative controls, no T-cell response to MERS coronavirus overlapping peptides. And the positive control are PCR confirmed cases from Saudi Arabia. And you can see most of them do have T-cell responses. And now here you have people from Nigeria. These are abattoir workers working with dromedary camels. You see some of them do have CD4 T-cell responses to MERS. Workers in the market without direct exposure to dromedary camels have no such responses. And other people in the same town had no T-cell responses. And if you look at CD8 T-cell responses, the same thing. So if you look overall, about 1/3 of those camel abattoir workers working closely with camels had evidence of specific T-cell responses. And we also confirmed these T-cell responses by looking at accessory proteins of the MERS coronavirus, which are unique to MERS coronavirus. So these proteins are not found in either SARS-CoV-2 or any of the other seasonal coronaviruses. So now this is one study, but it really is a study that needs to be repeated and confirmed to be sure, of reproducibility. Right. So we have, as I said, with antibody and with T-cell responses, shown evidence that infection is taking place in Africa, outside the Arabian Peninsula. So why is it that are there changes in the virus that makes it less severe for humans? Is that the reason why human cases are not being reported? And in collaboration with a number of groups in Morocco, Egypt, Ethiopia, Kenya, Nigeria, Burkina Faso, we ourselves have done a number of field studies in camels and got camel sequences and camel viruses, that is MERS coronavirus from camels and virus isolates from camels. And other people have also got data, particularly from the Erasmus group in the Netherlands and Christian Drosten and his group in Germany. And here you can see in this phylogenetic tree, when you put all these virus sequences together, the initial MERS coronavirus that was reported in Saudi Arabia falls into a separate clade, clade A. And most of the more recent coronaviruses, MERS coronaviruses in the Arabian Peninsula fall into this clade B here, all of these here. From Saudi Arabia, United Arab Emirates, Kuwait, Qatar, etc. But all the African viruses grouped separately in a separate clade, which is clade C. And some of these African viruses also have deletions in different parts of the genome. Now, as part of these studies, we were also able to isolate viruses from Saudi Arabia, Egypt, Burkina Faso, Nigeria, Morocco, Ethiopia, and Kenya. And so we decided to try to compare them phenotypically. They're genetically distinct, but still, even genetically distinct, they're more than 99% nucleotide identical, right? So we wanted to see if there's any biological difference in these viruses. And to do this, we have a very nice system of human respiratory tissues, which we can maintain in culture. These are not cell lines. This is actual human respiratory tract tissue. Most of it is coming from respiratory tissue removed during surgery in patients who are having lung resection for various reasons. And we take the healthy part of the lung, which is removed and thrown away, and we grow it in culture. We can do that for a number of days. And here you can see infection with MERS coronavirus. This red signal is showing immunohistochemistry for MERS coronavirus. So using this system, we can look at the replication competence of viruses from the Arabian Peninsula with that from Africa, Burkina Faso, Ethiopia, Kenya. And this is in the bronchus. You can see these viruses replicate less efficiently compared to the Saudi viruses, and also in the lung, the same, less efficient. And I'm not showing you the data, it's there in the paper, but the Nigeria and Morocco viruses also are behaving like this. That is, they have less replication competence. We looked in a mouse model, and we see the same thing. So in mouse lung, you find that the viruses from Saudi Arabia have higher viral titers in the lung compared to viruses from Africa. So it does appear that the viruses from Africa appear to have lower replication competence in the human respiratory tract. They're genetically distinct. And of course, we still don't know what are the genetic determinants of this lower replication competence. But this may lead to lower disease severity in humans. And maybe because of that, they're not being recognized. So this is one possibility that we are following up. But the important point to keep in mind is that these viruses from camels in Africa are extremely unstable genetically. And what I'm showing here is deletions in this case, the ORF3 accessory gene region. This is the ORF4 gene region. So this is the full genome that you see for that gene. But you can see many different viruses have deletions in these areas. So the virus is genetically unstable. And this type of situation is extremely dangerous because you can have quite unexpected changes in virus tropism, species adaptation, and severity as well. The other question, of course, is what? So as I showed you, there seems to be a difference virus genetically and in terms of phenotype or replication competence in humans. But what about the replication competence in camels between the African viruses and the Arabian Peninsula viruses? And this is a study we did in collaboration with a group in Barcelona in Spain, who have a BSL-4 facility where they can actually use alpacas, which are camelid species, because camels are too big to put in BSL-4. And they compared the Egyptian virus with a Middle East virus, and essentially show that also in camels, the African viruses seem to have lower replication competence in camels. Now, so then the question is, so this may explain why the clade C viruses from Africa have not been detected so far in the Arabian Peninsula. And when I say surprising, it is surprising because there is a lot of movement of camels from the Horn of Africa here into Saudi Arabia, particularly for meat. So the live camels are moved, are shipped over and slaughtered in the Arabian Peninsula. So the reason why clade C has not got established in Saudi Arabia may be because the clade B, which is already present, is better fit for in camels as well. So it doesn't allow clade C to get a foothold. But of course, this means that if by some chance the clade B viruses get into Africa, it can probably actually outcompete the existing viruses, and it may cause bigger problems. Right, so coming back to the human issue, so we have talked about humans are being exposed to infected animals in Africa. Behavioral factors suggest a lot of exposure. There is evidence of infection taking place. We said the virus genetics is slightly different and the phenotype is different, but still infection is taking place. So why human cases have not been recognized? I mean, it could be that some of these cases may be milder, but it also could be because many healthcare facilities in Africa do not even think of MERS. They don't test for MERS when you have patients with severe respiratory disease. So it just may not be being recognized. So just to conclude this part, as I said, MERS coronavirus can cause large outbreaks of severe human disease, so far limited to the Middle East in humans, or at least zoonotic diseases. Dromedary camels are the zoonotic source. MERS coronavirus is enzootic in dromedary camels in most parts of the world, including Africa, the Middle East, and Central Asia. MERS coronavirus infections are occurring unrecognized in Africa. I told you about the genetic and phenotypic differences, and this may be a reason why it is not recognized, but the virus is genetically unstable and is still a cause for concern. Now, of course, closer to your part of the world, again, the virus has been detected serologically and by PCR, including in Pakistan, and this includes work done by Massab and his group as well. But there is no genetic data available from these viruses, so it is extremely important, I think, to try to just like I explained the studies in Africa, to understand better the MERS coronaviruses in Central Asia. Now, I will very quickly go through this other part of this work, which is something that we have done. So one of the questions one of my PhD students were trying to address is there, so the virus is repeatedly jumping from camels to humans. Is there evidence of genetic adaptation in humans? So this is what he did. So he collected all the virus sequences available in public databases from camels and from humans, and essentially looked right through the genome to identify mutations that are more frequently found in humans than in camels. And he did find some. So this is in this particular gene called NSP6 with of the virus. And so we looked at the raw data set. We have also looked at the adjusted data set to exclude oversampling due to outbreaks, etc. I won't go into the details of it. But again, we come up with the same mutation in the same part of the genome in NSP6. And if you look at this on a phylogenetic tree, you can see so this is the clade B viruses. In orange are camel sequences, in blue are human sequences. And you can see that repeatedly from camels to humans, when the virus seems to jump, the mutation arises. So these triangles are where the mutation is present. Occasionally you see jumps to humans without the mutation, like here and here. but most of the time the virus is acquiring the mutation. So it seems to be an adaptive mutation. The virus is trying to adapt to humans. And this is the protein itself, NSP6, and the mutation is present here in the cytoplasmic domain. So very quickly, my student looked again using reverse genetics, so using a clade B virus from South Korea. He then mutated that back to the camel amino acid, so he can compare identical virus genome. The change is only in this one place in the whole genome, in this NSP6. So you can see the plaque size. So the virus with the human mutation has larger plaque size compared to the camel amino acid. And if you look at replication competence, again, the human virus is replicating better. And if you look at competition experiments, that is, if you mix the human and the camel virus, if you like, and allow it to replicate in this case in vero cells, always the human virus wins out, meaning it is able to replicate better than the camel virus. And similarly, we did this also in the mouse model, just like I told you in the African studies. And again, that single amino acid change you can see has a huge effect. So this is more than 1 log difference in titer in the mouse lung. And in these ex vivo cultures, again, we can see in the nasal and the bronchus, again, that single amino acid change is allowing the virus to replicate better. Now, we know that NSP6 has a number of biological functions like innate immunity and double membrane vesicle formation, etc. We have tried to identify what our particular mutation is doing, but up to now, the mechanism is still unclear, although the fact is very clear. That is, when the virus jumps to humans, the virus is trying to adapt to humans. So as I said, we have this recurrent mutation when the virus jumps from camels to humans, and using reverse genetics and isogenic viruses, we were able to show this gives selective advantage in replicating in human cells, but the functional mechanisms remain unknown. So just before I end, I'd just like to point out a number of surveillance tools that are available in the WHO and the FAO website on human surveillance, on camel surveillance, which is available, and you can use any of those if you're interested in doing any studies in Pakistan on MERS coronavirus, which I think is extremely important to do. And just to summarize the whole talk, I think I have showed you that the MERS coronavirus continues to evolve in dromedary camels with fairly unpredictable consequences. The clade B viruses, when it jumps to humans, you can see the virus is trying to adapt. And in future, we may have more adaptations which may allow the virus to transmit more efficiently. The clade C MERS coronavirus is genetically unstable, which is the virus found in Africa. It is infecting humans in Africa, but it's still is not suspected and unrecognized because there's minimum surveillance or awareness. As I summarized, it is genetically distinct and phenotypically distinct, but the virus is continuing to evolve and needs continued surveillance and risk assessment. And in particular, I think, as I pointed out, there is need for studies on MERS, particularly to characterize the virus in camels in Central Asia, because, as I said, we still don't know anything about the virus sequence or the phenotype, that means the virus behavior, biological functions of the virus that is circulating in camels in Central Asia, including in Pakistan.
Yeah.
So just to finish, this is some of the group in Hong Kong. And very importantly, as I said, we have collaborated with many groups in Nigeria, Ethiopia, Kenya, Burkina Faso, Morocco, Lebanon, etc. And I also mentioned, ongoing studies we have with Dr. Massab in Pakistan. Thank you very much. I'll be very happy to take any questions.
Thank you so much, Dr. Malik. I think it was an excellent talk and really informative I'm sure participants have learned a lot about MERS-CoV. So there are a couple of questions. So one question is, how long do neutralizing antibodies persist in camels post-infection?
Right. So neutralizing antibody in camels post-infection does seem to be very long lasted. And also the antibody formation is much more efficient than in humans. I told you that in humans, mild infection doesn't give rise to antibodies. But in camels, it does even I mean, as you know, the infection in camels doesn't lead to any serious disease, just a mild kind of rhinitis type of thing in camels. But they get they get good antibody response in following that. And now it's not very easy to be absolutely sure how long the antibody response lasts, because in the field the camels are getting re-exposed and reinfected, right? And in studies we have done in Saudi Arabia in collaboration with our colleagues there, where we followed the same herd over and over again by sampling uh, every 4 months or 5 months, over 2 or 3 years. We can see the camels who got infected, after some time they can get reinfected again. So because of that, we don't know whether the antibody persistence is because of repeated infection, or whether the antibody is kind of persisting. If there was no reinfection, whether the antibody would persist as well. But the best sense I get is that the antibody in camels is quite a sensitive indicator and a long-lasting indicator of infection. Because just to give you one very unusual example, you remember I told you that the studies done in Canary Islands by Marian Koopman's group. So in Canary Islands, there was no evidence of virus circulation in Canary Islands. If you remember, Canary Islands is off the west coast of Africa, right, far away. Um, so those camels had been transported from somewhere in Africa to the Canary Islands. So clearly they were antibody positive, even though they were not getting reinfected. So I think in conclusion, it means that the virus antibody response in camels is long lasting.
Thank you, sir. So there is another question. You have determined differences in MERS-CoV clade B and C in infecting lung tissue samples. Have you tried testing lung tissue collected from different regions, Saudi versus African?
Lung tissue from humans, you mean, or camels,
Yeah,
or
uh,
humans?
yeah.
Yeah, yeah. No, no. I mean, because as you might know, it is not very common to have post-mortem or autopsy studies in patients. That is not culturally very acceptable in parts of Saudi Arabia. So it's very difficult to get access to the virus in the lungs. But your question is quite important in a sense, because one would expect further adaptation taking place in the virus in the lungs, as the virus is more and more, replicating more and more in the human lung. One would expect that you're getting more and more adapted to humans. Most of the virus sequence data we have is either from the nasopharyngeal swab, or, of course, sometimes, occasionally, from bronchoalveolar lavages. So, in answer to your question, it's an important question, but we don't know. We don't have that type of data.
Okay. So one question I have, Dr. Malik, do you think are there any promising vaccines or treatments that are under development, and what are the challenges bringing them into the clinical use?
Yes. So treatments now. So again, this is the issue, because in order to show that a treatment is working, you have to do randomized clinical trials in humans, right? And it's very difficult to do because so far the number of cases, it's kind of difficult to set up. But there has been a control trial set up in Saudi Arabia, The particular treatment that was tried is a combination of what is it? It's Calitra. So it's one of these HIV drugs, which is lopinavir and ritonavir, because in the test tube or in vitro, it does seem to have a inhibiting effect on the virus, just as it also has an inhibiting effect on SARS-CoV-2, right? And if you remember, this was also, people were using it during SARS-CoV-2 as well. But when the randomized clinical trial data came out, and this was the large clinical trials that were carried out in the United Kingdom, essentially, it didn't seem to confer any benefit, right? Now, in the Saudi Arabian control studies, also in the case of MERS, there has been no benefit seen with that particular combination. Now, there have been some other anecdotal studies, small sample sizes, etc., but not properly controlled. So people have used convalescent plasma, again, without any convincing benefit. Convalescent plasma meaning plasma taken from people who have recovered from MERS-CoV-2. So that means having antibodies given to patients who have MERS-CoV-2. But the limitation of that study is that they did not actually titrate the level of antibody. If you remember, I told you that some patients don't make any antibody, right? So just because a patient has MERS and you take blood from them or plasma from them and transfer it to somebody else, it may not even, you may not have any antibodies for it to work. So passive antibody is another option that I think possibly needs to be used. And we actually did some studies in mice where we transferred antibody from camels. Camels have high titer antibody. We transferred it to mice, and we have controls without transferring antibody. And clearly, the antibody did seem to reduce severity of disease in the mouse model. But as I said, human clinical trials, no evidence. Prednisolone or corticosteroids, which, as you know, there is evidence for its use in SARS-CoV-2, but in oxygen-dependent patients. Now, in MERS, that has not been shown to be of benefit. But again, we don't know whether that is because these patients were already too sick by the time they got it. And these are very small clinical trials, right? So I think the lesson that we learned from COVID-19 is that you really need large sample size for human clinical trials to get reliable data. So in answer to your question about treatment, we don't have any good treatments, specifically antiviral wise. I mean, of course, ventilation and oxygenation and all those supportive care is there. Now, in terms of vaccine, yes, indeed. I mean, you remember in COVID-19, one of the vaccines that was available was the adenovirus vectored vaccine, which came out of the Oxford group. Now, before COVID-19, they had already developed this same adenovirus vectored vaccine as a vaccine for MERS. And they had done phase 1 clinical trials with that vaccine. So this is why they were able to very quickly switch. When COVID came, they were able to switch the same platform to SARS-CoV-2. Now, of course, they are going back to MERS coronavirus, and they're trying to do clinical trials, at least for phase 1. And there's another vaccine for MERS, which is a vaccinia-based. You remember the smallpox vaccine, a vaccinia-based vaccine. But again, none of them have been shown to be clinically effective in MERS because obviously case numbers are so small. So that is very difficult to do a clinical trial in humans. So that is the situation. So we don't really have good interventions, but there is a lot of effort being taken in that direction. And as you might know, MERS is one of the diseases that are high on the priority list for WHO and for CEPI, which is the Coalition for Epidemic Pandemic Preparedness Initiative for vaccine development. So there is a fair bit of work going on. Any other questions?
Yeah, so in the absence of specific treatment and vaccines, what are the most effective intervention strategies to prevent and control MERS-CoV transmission from camels to humans? And this is again a question posted by one of our participants.
Yeah, so I think basically trying to reduce spillover. Well, of course, in theory, the spillover is taking place at the camel-human interface, right? So one of the first things, say specifically for Central Asia, the question is whether there is spillover taking place, whether the virus that you're finding in camels in your part of the world, is spilling over or not. So in order to do that, you have to do a number of clinical studies or a number of studies, right? You have to, as we said, get at the virus, look at the genome of the virus, do the type of phenotypic studies that I described, and then also do studies in humans looking at seroprevalence and PCR positivity in people who are regularly closely associated with camels. So these camel herders, camel slaughterhouse workers, etc. So this type of systematic study is important to find out whether there is human infection taking place in your part of the world as well. And if there is, then one can think of ways to reduce exposure and risk, although it can be practically difficult to do. For example, in camel herders. And then at the other level, it is quite important to reduce the chance of human-to-human transmission in hospitals. Now, this is something that, of course, has been very successfully done in the Middle East, in Saudi Arabia and United Arab Emirates and Qatar and places, where with high awareness and patient isolation, rapid testing for these patients, any suspected patients as they come into hospital, so that they're isolated and kept in isolation without allowing transmission to take place, you can reduce the human-to-human chain of transmission, which is ultimately what will lead to the virus adapting and becoming pandemic. So that is something that, of course, can be done with awareness. But again, I think you have to start with doing studies of the local virus that is there in Pakistan and Central Asia, first and foremost. So there's a whole lot of things I think that needs to be done. Thank you.
Thank you so much, Professor Malik. I think we are coming to an end. I would like to thank you, sir, for taking time out and for presenting such an informative work here. I'm also extremely grateful to all the participants. I hope this was an informative session for you all. Just to let you know, sir, most of the participants, they are working in the National Public Health Laboratory Network of Pakistan. So I hope this was quite useful for them. So again, this webinar will be shared on our YouTube channel, Viral Insights. So we will share the link with you as well. So thank you so much. Thank you, sir. Take care. Bye.
Thank you. Bye.